Hello everyone. Today we are going to discuss about the topic gametogenesis in humans. We need to understand the meaning of the term gametogenesis. By definition, gametogenesis is the process of production of male and female gametes in the male and female reproductive organs respectively. Gametogenesis in humans can be of two types: spermatogenesis in males and oogenesis in females. We are going to divide the board into two halves and discuss each one. The schematic representation for each one will be drawn and discussed. Gametogenesis in humans. As I have already stated that it is of two types: spermatogenesis in males and oogenesis in females. Now, the process of spermatogenesis in males starts at puberty. It starts at puberty under the influence of the hormone GnRH, that is gonadotropin releasing hormone. Gonadotropin releasing hormone secreted by the hypothalamus of the brain. This hormone acts on the anterior pituitary and the production of it acts on the anterior pituitary to produce two hormones: luteinizing hormone and follicle stimulating hormone, that is LH and FSH. LH is termed as luteinizing hormone and FSH is termed as follicle stimulating hormone. This luteinizing hormone or LH acts on the Leydig cells of male testis. Leydig cells of male testis and helps in the production of androgens, mainly testosterone. Whereas the FSH or follicle stimulating hormone acts on the Sertoli cells, which are also called the nurse cells, and helps in the production of ABP, also termed as androgen binding protein. Androgen binding protein that helps to maintain a very high level of testosterone in order to maintain the process of spermatogenesis. So this was the hormonal control, all about the hormonal control of spermatogenesis. We'll revise it once again. Spermatogenesis starts at puberty in males under the influence of gonadotropin releasing hormone, also called GnRH, which acts on the anterior pituitary, releasing two more gonadotropins, which are LH and FSH respectively. LH is luteinizing hormone and FSH is follicle stimulating hormone. LH acts on the Leydig cells of male testis and helps in the secretion of androgens such as testosterone, whereas FSH acts on the Sertoli cells, also known as nurse cells, and release the factor androgen binding protein, which helps to maintain a high level of testosterone in order to fuel the process of spermatogenesis. Now we'll discuss the schematic representation of this process in detail. As we have already stated that spermatogenesis in males starts at puberty. It starts with the spermatogonial germ cells. Spermatogonia, or also called the male germ cells. Spermatogonia, which are also called male germ cells, having diploid number of chromosomes, that is the 2n constitution. Now, these spermatogonial cells undergo mitotic division to produce two types of cells. What are they? Type A D cell, and another one is type A P cell. Now, what does D and P stand for? We need to know that. D cells are those cells of type A which have dark nuclei. So, D stands for dark nuclei, whereby P cells are those types of A cells which contain pale nuclei. So, P stands for pale nuclei. Right? Now, it has been seen that this type A D cells, they do not further undergo any active mitosis. So, what is their significance? They actually fuel the process of spermatogenesis, means they act as reservoir cells and they keep on supplying a continuous supply of spermatogenic cells. Okay, so these actually are the reservoir cells which will fuel the process of spermatogenesis by producing more and more spermatogonia. Those are required for initiating the process of spermatogenesis. So, what is the fate of the type A P cell then? The type A P cells are the actively dividing mitotic cells. That means these cells will be actively dividing by the process of mitosis, giving rise to another type of cells known as the type B cells. These type B cells actually are the precursors of the primary spermatocytes, means the type B cells will now undergo again mitotic division. These are all mitotic divisions, so I write here mitosis, here also mitosis. They will give rise to the primary spermatocytes. So, these cells are the primary spermatocytes. Obviously, as we all know that mitosis division is an equational division. Equational division means if we start with chromosome number 2n, that is diploid, then by the end of the division, the chromosome numbers will remain the same. That means mitotic division being an equational division, so till primary spermatocyte, the chromosome number remains same, that is diploid or 2n. Now, these primary spermatocytes then undergo the first meiotic division to produce the secondary spermatocytes. These cells are called the secondary spermatocytes. As I said, this is the first meiotic division. As we know that meiosis is a reductional division, so obviously the chromosome number from the primary spermatocyte to the secondary spermatocytes will be halved. That means the secondary spermatocytes will be having haploid number of chromosomes. Now, each secondary spermatocyte will undergo the second meiotic division to produce two spermatids which are also haploid. So, this is the second meiotic division. I am writing over here this side, there is no space over there. Second meiotic division to produce two, that is each one produces two haploid spermatids. So, from two secondary spermatocytes we get four haploid spermatids. These are called spermatids. Now, these spermatids are generally round, so they need to undergo a process of differentiation to produce the otherwise tadpole shape like spermatozoa. So, by differentiation process, these spermatids transform themselves into spermatozoa. So here is the process of differentiation, whereby all the round spermatids which are haploid are getting transformed into the whip-like, or you can say tadpole-like shape of the spermatozoa, like this, with a head, neck, middle piece and tail. Obviously, these are also haploid in number because we know that second meiotic division is equivalent to mitosis. So, since mitosis is an equational division, so second meiotic division is also equivalent to mitosis. That means N will remain N by the end of the division. Now, we need to understand two concepts. What is spermatocytogenesis and what is spermiogenesis? Spermatocytogenesis is the process from the spermatogonia, that is the male germ cells, production of the production of the spermatids, the haploid spermatids. That means the process of formation of haploid spermatids from the diploid spermatogonia male germ cells is termed as spermatocytogenesis. So we are going to write here spermatocytogenesis. That means this is the process of formation of spermatids from the spermatogonia. Okay. Now, another process is known as spermiogenesis. What is spermiogenesis? We'll sum up the definition here. Spermiogenesis is the transformation of the spermatids into spermatozoa. That means this particular phase is termed as spermiogenesis. So we'll write here transformation of the spermatids into spermatozoa is known as spermiogenesis. So this was the overall schematic representation of the process of spermatogenesis in males, which include two basic processes, spermatocytogenesis and spermiogenesis. So here we'll understand a particular thing, that from one primary spermatocyte, how many spermatozoa which are functional we are getting. By the end of the process, from one primary spermatocyte, 4 functional spermatozoa are derived. Next, we move on to the process of gametogenesis in females, which is known as oogenesis. There is a vast difference between these two processes, that is spermatogenesis and oogenesis. First, we will draw and discuss the schematic representation on the board. After that, at the end of the discussion, we are going to sum up what are the differences between these two processes. Now, coming to the process of oogenesis in females. In oogenesis in females actually start even before birth. That means it starts during the fetal developmental stages, or we can say embryonic developmental stages. That means when the embryo is still within the mother's womb, at that time, when the embryonal, that ovaries are formed in the embryo, at that time itself, the number of oogonial cells are already fixed. They cannot later be added to the process. Okay? So this process starts even before birth. So during the fetal life, so I will write here fetal life. These are the precursor, these are the cells, that is the oogonial cells, oogonia. Obviously, they are diploid, that is having chromosomal constitution 2n. Now, these oogonial cells undergo mitotic differentiation like this to produce primary oocyte. Okay. Now, these cells are known as the primary oocytes. Primary oocytes are also 2N because this process is mitosis. Oogonial cells are undergoing mitotic differentiation. So process is mitosis. Hence the chromosome number becomes same, remains same, that is 2N remains 2N. Now what happens to these primary oocytes? Generally it has been observed that these primary oocytes enter the first meiotic division, but the division is not completed. That means what happens is the primary oocytes are temporarily arrested. They get temporarily arrested in the prophase 1 stage of meiosis. Now what is the significance behind this? Why do the cells, or why are these cells temporarily arrested in the prophase 1 of meiosis? They actually don't complete the meiotic division, because till the time the oogonial cells, which I have already mentioned that they are formed in the fetal ovary itself even before birth. So by the time the girl reaches puberty, she cannot conceive a child. She is not reproductively capable of conceiving a child. That is why it is the nature's creation that the primary oocytes are arrested in the first prophase 1 of meiosis. That means until and unless the girl reaches puberty by the age of 13 or 11 to 13 years of age, that she becomes reproductively capable of bearing a child, because at that time the primary oocytes will start their first meiotic division. So till then they are temporarily arrested, they grow in size, DNA duplication occurs. Now here we write birth, childhood, that is the phases in the woman's or the girl's life, childhood, and then we have puberty. Here I would like to take a contrast, come back to spermatogenesis once, and as I said that spermatogenesis process in males started at puberty. So I will just write here at puberty. But we can see very well that oogenesis process has already started during the fetal developmental stages itself. But by the time the girl reaches puberty, the number of primary follicles have already decreased in number. Now you need to know what are primary follicles. The primary oocytes, they are enclosed by, suppose this is a primary oocyte, it will be enclosed by a single layer of granulosa cell and form a primary follicle. Primary follicles. Now these primary follicles are enormous in number during the fetal developmental stage. But as soon as the child is born, the female is born, at that time the number of primary follicles reduce in number to a great extent. By the time the female reaches puberty, 11 to 13 years of age, the primary follicles have reduced so much in number that they are now confined to only 60,000 to 80,000 primary follicles. Right? Now what happens? So till here there is no meiotic division. The meiotic division, first meiotic division will start at puberty, whereby the primary oocyte now divides into a very small, divides unequally to produce a very small cell known as the first polar body, and a bigger cell known as the secondary oocyte. And as I have already mentioned that this is the first meiotic division. That means this meiotic division is completed prior to ovulation. You need to remember one thing, understand a thing, that the first meiotic division in oogenesis is completed prior to ovulation. Now what does that mean? We need to understand what is ovulation first. What is ovulation? If we study the menstrual cycle, which lasts for 28 days, starting from day 1 of menstrual cycle till 28 days, that is the period of span of menstrual cycle. Okay, menstrual cycle itself lasts for 3 to 5 days, but if we study the cyclical changes that occur over 28 days, then at that time we see that starting from day 1 of the menstrual cycle till day 14th, by the 14th day, there is an LH surge, means under the influence of too much of luteinizing hormone, the fully mature Graafian follicles rupture. To release the secondary oocyte is termed as ovulation. But the primary oocytes undergo the first meiotic division even before ovulation. That means the oocyte which is present inside the primary Graafian follicle is not a primary oocyte. It has already undergone the first meiotic division before ovulation and has transformed to the secondary oocyte. So now what happens? The second meiotic division in case of oogenesis is not completed in the ovary. Why? Because after this ovulation has occurred and the secondary oocyte is already outside the ovary, it has come out. So where does the second meiotic division occur? It occurs within the oviduct. Okay? So now what happens? This secondary oocyte now undergoes a second meiotic division. Second meiotic division to produce again a very small size cell known as the second polar body and a large cell known as the ooted. Now what happens? This ooted undergoes differentiation to produce ovum. Now what will be the chromosomal constituents of, till the primary oocyte we know 2n, so what will be the chromosomal constituents of the secondary oocyte? Obviously meiotic division is a reductional division, so chromosome number will be halved. So secondary oocyte will become n, first polar body also haploid n, then again the second meiotic division is again a reductional division but which is equivalent to mitosis, hence this n will remain n, that is ooted is haploid n and a second polar body n. Now ooted undergoes differentiation to form ovum which is also n, that is the female reproductive structure is termed, the female gamete rather is termed as ovum n. All these events, that is after the first meiotic division, from here all these events occur in the adult reproductive life. Okay, so this was the entire process of oogenesis in females. Next, our topic of discussion will be to summarize or sum up the differences between these two processes, spermatogenesis in males and oogenesis in females. One by one, we will see the differences. First and foremost difference, as it is very obvious, the definition. Definition-wise, spermatogenesis is the production of sperms in males. Oogenesis is the production of ovum in females. First difference. Later on, we'll jot down the differences on the board. Now, what is the second difference? Spermatogenesis process starts at puberty, whereas oogenesis process starts even before birth during the embryonic developmental stages. Third difference, during the process of spermatogenesis, from one primary spermatocyte, at the end of the process, we get 4 functional spermatozoa. But during oogenesis, at the end of the process, from one secondary oocyte, we get only 1 functional ovum and 2 to 3 polar bodies. In here you can see first polar body, second polar body and 1 ovum. So 1 ovum and 2 polar bodies. Now why do I say 2 to 3 polar bodies? Because in some cases it may happen that the first polar body also undergoes division to form 2 more polar bodies. But this is optional. This doesn't happen every time. So may or may not divide. The first polar body may or may not divide. If it doesn't divide, then by the end of the process we get 1 functional ovum, that is N, and 2 polar bodies. And if the first polar body again divides into 2 polar bodies, then by the end of the process we get 1 functional ovum and 1, 2 and 3 polar bodies. Okay, understood? That is why I said that 2 or 3 polar bodies. Now, one more thing is there, one more difference is there, that is the cytokinesis in case of these will result in the production of 2 equal sized cells. See, from the primary spermatocyte, the secondary spermatocytes are also equal sized cells. But here in case of oogenesis, the cytokinesis in the primary oocyte will result in an unequal division. See, this is first meiotic division is unequal division with the production of two very highly unequal cells. One cell is very small, having little bit of cytoplasm known as the first polar body, and another is very big, having larger amount of cytoplasm called the secondary oocyte. Again, the second meiotic division in case of spermatogenesis, see this one, second meiotic division, this is also equal division. That means cytokinesis is resulting in equal size cells. But here you come to oogenesis, the second meiotic division, this one again produces two highly unequal cells. One is a very small second polar body and another is a larger ootid. After differentiation, from the spermatids, 4 functional spermatozoa are formed, whereas in case of oogenesis, after differentiation, the ootid transforms itself into a single haploid ovum. Okay, so that was all for today. Thank you so much.